Today, I'm going to show you how to make a Winogradsky column, named for the Russian scientist Sergei Winogradsky who invented the method. Building a Winogradsky column is a great way to observe microbial diversity in nature, and it is super easy so you can try it at home. The basic idea is to collect a mud sample and place it in a clear column like a plastic bottle for observation, and then watch the microbial life flourish over time. So let's get started. First, we'll need to find some mud. You might be able to find this in a puddle in your own backyard, or maybe you'll need to take a visit to a local stream, pond, or river. Dress in something you are willing to get a little dirty, wear gloves to keep yourself safe, and bring along a shovel and a bucket. For my column, I went to a local park and found a nice puddle. Use your shovel to begin scooping out mud, collecting it from just below the surface of the water. Get just a bit more mud than you expect that you'll need to fill a 1-liter bottle, since you will later have to remove some objects like rocks and plant matter. Now we have our mud and we can return home to start building our column. Now let's gather our materials. We'll need a clear 1-liter bottle, 2 large bowls, 2 large spoons, a dark pen or marker, a pair of scissors, plastic wrap, a rubber band, a ruler, a funnel, a light source such as a window or a desk lamp, 1 egg, a workspace, and paper of some sort. I used 2 bottles for mine, but you can make as many or as few as you would like. First, remove the bottle wrapping so you can see clearly through the column. Then, cut off the tops of the bottles and use a ruler to mark a point about 1/4 and 3/4 of the way up from the bottom of the bottle. Next, cut up about 1/4 of paper into thin strips and place 1 egg yolk into a bowl. Set these aside for now. The egg will be a source of sulfur and the paper will be a source of carbon. These resources are necessary for the metabolism and survival of some microbes, while other microbes may simply obtain the needed resources from the air or organic matter in the environment itself. Remember to wear gloves when working with mud. Begin scooping mud into one of the large bowls, sifting and removing any large objects like stones or clumps of plant matter. Stir it to the smooth consistency of a milkshake. Next, add some of this mud into the second bowl, to which you'll add the egg and paper. Stir these materials together in the mud and then add this mix to the 1/4 line in the bottle. Next, add the mud from the first bowl to the 3/4 line. And finally, add water reaching to about 1 inch below the top of the column. Now we can cover the bottle with a layer of plastic wrap and secure it with a rubber band. And that's our completed Winogradsky column. We'll want to ensure that it receives light, so find a spot with sunlight or place it beside a desk lamp. Leave it there for about 6 to 8 weeks to give it enough time to establish, and then observe what grows. Here is a well-established Winogradsky column made by the microbiology students in the Microbial Diversity Lab Mic 105L. We can expect to find photosynthetic microorganisms such as cyanobacteria at the top layer of a column, since here they have access to the most sunlight and to the air, from which they need carbon dioxide. We are also more likely to find microbes that do aerobic respiration near the top of the column, and microbes that do anaerobic respiration nearer to the bottom, where they have minimal access to oxygen. Generally, we might find phototrophs and autotrophs nearer to the top, and chemotrophs and heterotrophs nearer to the bottom. If you take multiple samples from various locations, you'll also be able to observe the differences in microbial ecology corresponding to different areas. I hope you enjoyed this tutorial. Now you can go make your own Winogradsky column and learn about the diversity of microbes in your environment. Thank you.